There are three main processes in urine formation. First, ultrafiltration. Second, reabsorption. Third, secretion. First process, the ultrafiltration of blood that takes place at Bowman's capsule. It's known as ultrafiltration because it filters out smaller molecules than red blood cell and plasma protein such as amino acid, glucose, sodium, potassium and chloride ion and become the part of the filtrate. There are factors contribute to ultrafiltration. First, the hydrostatic blood pressure in the glomerulus higher than in other capillaries due to the smaller diameter of afferent arteriole than afferent arteriole. Second, the large surface area for filtration provided by highly coiled glomerulus capillaries. Third, the high permeability of the glomerulus. Second process of urine formation is reabsorption. As the glomerular filtrates flow through the nephron, various substances move out of it. Most reabsorption takes place in the proximal convoluted tubule. In the proximal convoluted tubule, selective reabsorption occurs. All glucose, amino acid, vitamin, and many inorganic ions are reabsorbed back into the blood. Substances are reabsorbed by both active and also passive process. The third process of urine formation is the secretion. Secretion is the process by which substances move into the proximal, distal, and collecting tubules. As the filtrate flows from the cortex to the medulla in the descending limb of the who, water leaves the tubule by osmosis because the interstitial fluid is the hypertonic. The water moves straight into the capillaries surrounding the loop of Henle. The loss of water concentrates sodium ion and chloride ion in the descending limb. The concentration of the filtrate at this highest at the bottom of the loop of Henle. The ascending limb is relatively impermeable to water. As the filtrate flows up the loop of Henle, the sodium ion and chloride ion diffuses out from the thin segment of the ascending limb to the interstitial fluid in the medulla. In the thick segment of the ascending limb, the sodium ion and chloride ion are actively transported into the interstitial fluid of the renal medulla. The filtrate entering the distal tubule is hypotonic to the peritubular fluid. As the filtrate descends again toward the medulla in the collecting duct, water is extracted by osmosis in the hyperosmotic interstitial fluid. This concentrates salt, urea and other solids in the filtrate. Some urea leaks out of the lower portion of the collecting duct, contributing to the high interstitial osmolarity of the inner medulla. The urine in the collecting duct becomes progressively more concentrated as it moves into the pelvis. the model of the kidney of human being. This structure is the kidney of a human. Here is the cross-sectional area of the kidney. These are the main structures. This structure is the renal medulla, and here is the renal cortex, while here is renal pelvis.
Ada sebuah cerita tentang formation of urine di dalam kidney kita dengan 3 proses sahaja: ultrafiltration, reabsorption dan tubuh secretion. First, ultrafiltration. Kita tapis bahan berguna ke dalam momen kapsul dengan relative pressure tinggi. Melalui glomerular sel momen kapsul.
This is surely not what you thought it would be. I'm gonna tell for the next day. Reabsorption bila lalui TCP, bahan diserap semula ke salur darah. Air masuk secara osmosis, sodium dan klora akan dikeluarkan.
Sekarang tiba masa untuk tubuh secretion. Potasium, amonia dan bahan toksik keluar become urine.